Hi guys, welcome back to my channel. It's Doc Janelle. The subject at hand right now is Human Anatomy and Physiology. So welcome to our pre-lab discussion. For this video, we are going to be talking about lab exercise number 5 or transport through cell membranes. So what cell do you see here in this image? This is actually an example of a group of red blood cells. Now, as we can see in this image, each individual cell maintains its shape. Each individual cell is in balance or homeostasis with its environment. Basically, each individual cell is able to function well and interact properly with its environment because there is homeostasis between the amount of fluid inside and outside the cell. And the part of the cell that maintains this homeostasis between the fluid in and out of the cell is none other than the cell membrane. So by definition, the cell membrane is defined as the barrier against movement of water molecules and water-soluble substances between the extracellular and intracellular fluid compartments. In order for us to better understand what it means, it's only imperative for us to look at what the cell membrane looks like. So they say that this kind of diagram is called the fluid mosaic model. It is called in such a way because it shows us that the cell membrane is composed of a double layer. So there is a layer on top and a layer at the bottom. This double layer is actually composed of phospholipid with a hydrophilic end made of phosphate. So I just want to point out to you that the phosphate here are the big flesh-colored circles that you see here on top. And we also have that at the bottom. That's why it's a double layer, one layer on top, one layer at the bottom. So attached to your phosphate heads are your lipid portions, which are those extensions, thread-like extensions, no, coming from your phosphate heads. So this phosphate heads, they are described to be hydrophilic, which means that they can come in contact with water, while the lipid portion, the ones in the middle, are hydrophobic or they are afraid of water. So in order for us to better understand, I would like for you to imagine a sandwich. Imagine a sandwich with bread on top, ham in the middle, and then another bread at the bottom. So in this diagram, think of our hands as water. So when we want to eat the sandwich, we definitely have to hold the sandwich on the top bread and at the bottom bread. So this means that the top bread symbolizes the phosphate. It can come in contact with water, which is our hands, right? We can touch the phosphate part, or water can touch the phosphate part of the cell membrane. But our hands should not touch the ham in the middle or the lipid portion in the middle. So hydrophilic means it loves water or it can interact with water, and hydrophobic means it hates water or it does not mix or come in contact with water. Also, another thing that you see here, if you can follow my pointer, are these big structures here, and these are what we call as proteins. Now, proteins, their job is to act as a bridge to allow substances from outside to be able to enter and to allow substances inside to be able to exit the cell as well. So there are two types of proteins that we see here, those that protrude or pass through the cell membrane, and those are what we call as integral proteins. And we also have proteins that are only found on one side, and these are what we call peripheral proteins. The last structure that we see here are these red antenna-like structures. These are your carbohydrates, and they function to act as glue for cell adhesion and also to act as antennas for cell communication. Now, once we talk about transport, two terms come into mind: active or passive. In in its basic essence, we call a transport as passive when it does not use energy to transport substances from one place to another. And we call it active transport when there is now a use of energy in the form of ATP. Here is a better diagram showing us the different types of cell transport. So, diffusion is actually an example of passive transport. So we see here that in diffusion, there are two types: simple and facilitated. We'll go to that later. And for active, again, we said that there is a use of energy and a use as well of carrier proteins. More about that in a while. Now, I want to show you this image which shows cells having natural movement. So this is what we call Brownian motion. Cells have a naturally occurring, constant, and random movement. And because ang mga cells natin, kusa silang gumagalaw, they move on their own, this triggers cell transport. Okay? Now, another factor that we have to understand is the concentration gradient. So the concentration gradient means that there is a difference in the concentration between two areas. I want you to follow my pointer here. We have here a jar of water. And in one side of that jar, we place, let's say, coffee granules. So the coffee granules are concentrated on this area. And because of that, we were able to create a concentration gradient, an area of higher coffee granule concentration and an area of lower coffee granule concentration. So in a fluid compartment, if there is a difference in concentration between two areas, as seen in this image, there is a concentration gradient. So, because we can see a higher concentration of red dots on the left side compared to the right side, these red dots will activate their Brownian motion. So, gagalaw yan. And because of that random movement, it will move from an area of higher concentration to areas of lower concentration. And the goal of that movement is for it to be equally distributed, or it wants to reach homeostasis. So, ganun pala yun, no? When we have an area of higher concentration and lower concentration, the cells or the solutes with their Brownian motion will move from area of higher concentration to area of lower concentration until maging equally distributed yung concentration nila, or until ma-reach natin yung homeostasis. So because of Brownian motion and concentration gradient, we now have the first type of passive movement, which is diffusion. Diffusion is actually the simplest example of passive transport. It is the movement of particles from area of higher concentration to lower concentration. So let's look at this GIF on the side. Ayan. So it started out with an area of higher concentration and an area of lower concentration. And because these red dots or these solutes have a Brownian motion, it will continually and randomly move until they become equally distributed. Look at that, no? Ayan. So now equally distributed na sila. Also, even if we put a barrier in between two areas, in diffusion, they can freely pass through the cell membrane as long as it follows the concentration gradient. Now, that is very important and I want you to remember that. Sa diffusion, kahit may cell membrane tayo, it will move from an area of higher concentration to lower concentration. So in this second GIF, there is higher concentration on the left, and because of Brownian motion, nag-move sila until maging equally distributed sila. Now, we mentioned earlier that there are two types of diffusion, simple and facilitated. Ano yung difference ng dalawa? When we say simple diffusion, cells or solutes can easily pass through the cell membrane, or they can use a protein channel so that they can pass through the cell membrane as long as it follows the concentration gradient. Now, facilitated diffusion naman uses a carrier protein. Kung kanina, protein channel or channel protein, in facilitated diffusion, it uses a carrier protein. And I want you to remember the term conformational change. What does that mean? It means that in facilitated diffusion, pag gustong pumasok ng molecule na ito, papasok ng cell, it has to attach to the binding point of the carrier protein. So once it attaches to the binding point, look at the shape of this protein channel, or this carrier protein rather. Once it binds to the binding point, there will be a conformational change, or a change in the shape and appearance of the carrier protein. So look at this. Una, sa taas siya naka-open. But when it bound to the binding point, nag-close yung taas at yung ilalim ang nag-open. Still without the use of energy. So in facilitated diffusion, the transported molecule binds to a certain area in the carrier protein, and the carrier protein undergoes conformational change, or a change in shape, to allow the molecule to pass through. So that is diffusion. Another thing before we move on, or that I want you to remember about diffusion, is that when we talk about diffusion, we are talking about the concentration of solutes. Okay? Concentration of solutes. So we want the solutes to move from an area of higher concentration to area of lower concentration. I had to emphasize that because we have another type of passive diffusion, of passive transport, which is called osmosis. So osmosis is defined as the movement of water across a semi-permeable membrane, still caused by a concentration difference of water. So when we talk about osmosis, it is the movement of water from an area of higher concentration to an area of lower concentration. So let's compare these two compartments here. The one on the left has more water molecules symbolized by the blue dots, while the one on the right has lesser water concentration. But there are more solutes that you can find here. So basically, what water wants to do by virtue of osmosis is to move from area of higher concentration to an area of lower concentration. So from left, the water molecules will move to the right to basically reach balance. Now, once we talk about osmosis, you will hear the word tonicity. And when we talk about tonicity, it refers to the concentration of solutes. Okay? Solute ang tinutukoy ng tonicity. Now, why did I have to mention that? Because if we want to understand osmosis, we have to talk about the different tonicity of solution. So here we have a test tube, and the test tube contains water. And the water is a hypotonic solution. Hypo means low. And again, tonicity means concentration of solute. So by definition, this solution has a low solute concentration. So if there are lesser solutes, of course, there are more water. So, or more water concentration. So this solution in the test tube, the water pa lang ang pinag-uusapan ko ha. The solution in the test tube has higher water concentration. So what we did is we placed a drop of cell in the solution. So yung cell natin, meron din siyang water sa loob, 'di ba? Meron siyang intracellular water or fluid sa loob niya. So we want to know how the water molecules will move So let us first determine where is the area of higher concentration of water. So saan mataas ang water? Inside the cell or outside the cell? So since there is higher water concentration outside the cell, by osmosis, the movement will be water molecules will try to enter the red blood cell. So the red blood cell will receive more water molecules and it will start to swell and eventually it will explode because of so much water inside of it, sasabog siya. And cell explosion is what we call as lysis. All right? Next, we also have what we call isotonic solution. This is a this is where there is equal water and solute concentration. So as we can see in this image, there are two water molecules outside, two water molecules inside, and there will be no movement anymore of water. Okay, kasi equal lang sila. But if we have a hypertonic solution, hyper means high, tonicity again is concentration of solutes. So high solute concentration. Kung marami ng solutes doon sa water, of course, there is lower water concentration. So again, we have a cell that we place inside the hypertonic solution. So let us determine anong mas maraming water molecule, inside the cell or outside the cell. So in this image, we can see that there are more water molecules inside the cell and lesser outside. So how will water move? It will move from inside the cell to the outer solution. So lalabas yung water molecule. So what will happen to the cell? The cell will get dehydrated, it will shrink, and the process of shrinkage is what we call as crenation. To basically summarize the three, ayan. Again, we have a hypotonic solution, low concentration of solutes, so there are more water molecules outside compared to the inside of the cell. So how will water move by osmosis from area of higher concentration to area of lower concentration? So it will enter the cell will swell until it will explode, and the explosion is called lysis. In isotonic solution, there is equal amounts of water outside and inside the cell. Thus, there will be an equal movement only. In hypertonic solution, there is more water inside the cell because there is high concentration of solutes outside. So, water will leave the cell until it becomes dehydrated and shrink, and that is what we call as crenation. Okay? Lastly, I also want to talk to you about filtration. Filtration is the movement of particles across a membrane from an area of high pressure to an area of low pressure. So if we have a mixture of water and sand and we put it on a funnel and we place a filter paper, this will be an area of high pressure. So water will want to move from an area of high pressure to an area of low pressure. So gravity here creates a pressure gradient causing molecules to move. Now, in order for us to better understand, I posted on your TLC a video of the lab experiment that we were supposed to do. But because of the recent um, events, we had to shift online. That's the end of my pre-lab discussion. Please watch the laboratory session. Goodbye.